The coelacanth. The year is 1938, December. A South African museum curator named Marjorie Courtenay-Latimer is picking through the day's catch at a dockside fish market in East London, on the edge of the Indian Ocean. She sees something that stops her cold. It is steel blue, nearly 2 meters long, covered in thick armored scales that look less like skin and more like ancient chainmail. She has never seen anything like it. Nobody alive has, because the last confirmed record of this animal was a fossil laid down 66 million years ago in rock that formed when the dinosaurs were still walking the earth. The fish she is looking at was declared extinct before the first humans ever stood upright. And here it is, in a fish market, bleeding on a pile of lesser catches, blinking in the South African sun. This is the coelacanth, and its existence, confirmed in that moment and every moment since, is one of the most unsettling facts in the history of natural science. The coelacanth's lineage is 360 million years old. To understand what that means, consider this. The last time this animal's body plan was new, there were no trees on land, there were no reptiles, there were no birds. The continents were arranged in shapes you would not recognize on any map, and yet the fish that Courtenay-Latimer found was built from the same blueprint. Same hinged skull, same paired lobe fins, same arrangement of armored scales. When she contacted ichthyologist J.L.B. Smith to identify the specimen, he later wrote that the shock of seeing it was almost physical. He recognized immediately that the coelacanth's closest fossil relatives had last appeared in the geological record 66 million years ago, at the same boundary layer where the dinosaurs ended. 66 million years of silence. Then a fish market in 1938. What makes the coelacanth genuinely strange is not just its age, it is its anatomy. The fins are the first thing paleontologists point to. They are lobed, supported by an internal bone arrangement that follows the same pattern as the limb bones of every four-legged land animal that ever lived, including you. One proximal bone, 2 distal bones, then digits. No other living fish carries this structure. When the coelacanth moves those fins, it does so in a cross pattern, the same diagonal coordination used by a dog walking, a human running, a lizard crossing a rock. Its relatives were, supposedly, the line that eventually crawled out of the ocean and became everything that breathes air on land. The coelacanth was right there on that branch, and then, apparently, it turned around and went back into the deep. The skull is even harder to dismiss. The upper and lower halves are hinged along an intracranial joint, allowing the top of the skull to swing upward when the animal bites, a feature absent in every other living vertebrate. And then there is the rostral organ, a structure in the snout unique to coelacanths among all known animals. It is thought to detect electrical fields. What kind, at what range, in what configuration, science cannot yet say with certainty, because the function of the rostral organ has never been fully characterized. It is a biological tool that we cannot yet read. When researchers sequenced the coelacanth genome in 2013, led by scientist Chris Amamaya, what they found added another layer of strangeness. The protein-coding genes of the coelacanth evolve at the slowest rate ever recorded in a vertebrate animal. Not slowly for a fish. Slowest of anything with a backbone ever measured. As if some mechanism inside this creature is actively resisting change. A second living population was confirmed in 1998 near Sulawesi in Indonesia, 10,000 kilometers from the first. A separate species, genetically distinct, living in the same depth range, built to the same blueprint. 2 populations separated by an ocean, both surviving 66 million years of invisibility. The scientific explanation is that the coelacanth retreated to deep cold water after the KPG impact and simply persisted there, where its slow metabolism and cave-dwelling habits kept it out of harm's way. Deep water refugia, low competition, a stable niche. It is a reasonable explanation, but it explains absence from the fossil record. It does not explain the rostral organ, or the limb-like fins of a fish that never became a land animal, or a genome so resistant to mutation that biologists have no satisfying account of why. Some researchers ask whether the coelacanth's frozen anatomy represents not an evolutionary failure, but a kind of optimum, a body so precisely calibrated for deep ocean existence that any change would have been a step backward. Others wonder what exactly that organ in the snout is sensing in 4 kilometers of darkness that we have not yet thought to measure. What did not survive 66 million years tells you something. What did survive tells you something else. From the impossible fish of the Indian Ocean, we move to something even older and even harder to explain. Because the next creature on this list did not survive one extinction event. It survived 5. It watched the dinosaurs appear. It watched them vanish. It kept going. And then it showed up on a modern beach and started bleeding blue. The horseshoe crab. Every May, on the beaches of Delaware Bay in New Jersey, thousands of creatures crawl out of the Atlantic in the dark. They are armored, helmet-shaped, trailing long blade-like tails, climbing over each other in the surf to lay eggs in the wet sand. From a distance, in moonlight, they look like a slow invasion from another era, which is exactly what they are. The horseshoe crab has performed this same ritual on these same tidal beaches for 450 million years. The last time it did this, there were no birds to eat its eggs, no mammals to watch, and no predator on land capable of recognizing what it was looking at. There was, in fact, almost nothing on land at all. The horseshoe crab is not actually a crab. It is more closely related to scorpions and spiders, a member of the subphylum Chelicerata, whose closest confirmed fossil ancestor, Lunataspis aurora, was described from rock in Manitoba, Canada, and dated to approximately 445 million years ago, a discovery published in 2008 by researcher David Rudkin and colleagues. 445 million years. That is not a misprint. In the time since this animal's basic body plan was established, the Atlantic Ocean has opened and closed at least once. The Appalachian Mountains rose to Himalayan heights and eroded back down. The continents reassembled into Pangaea and then pulled apart again into the shapes we recognize today. The horseshoe crab watched all of it. Its shell looked more or less the same throughout. It survived the late Ordovician extinction, when approximately 85% of all species on Earth disappeared. It survived the late Devonian extinction, when the oceans became so toxic that 75% of species died. It survived the Permian extinction, the worst die-off in Earth's history, the event that killed an estimated 96% of all marine species. 96%. Nearly everything in the ocean died. The horseshoe crab did not. It survived the Triassic-Jurassic extinction. It survived the KPG asteroid impact 66 million years ago, the same event that ended the coelacanth's presence in the fossil record and eliminated every non-avian dinosaur on Earth. 5 mass extinctions. The horseshoe crab passed through each one and emerged structurally unchanged. And then in 1956, a biomedical researcher named Frederick Bang discovered something about its blood that made the modern world sit up and take notice. Horseshoe crab blood does not contain hemoglobin. It contains hemocyanin, a copper-based oxygen transport molecule, which is why it runs blue rather than red. More importantly, Bang found that the blood's amebocyte cells clot violently and immediately, when they encounter bacterial endotoxins with a sensitivity capable of detecting contamination at a concentration of 1 part per trillion. 1 part per trillion. No synthetic test yet developed has fully replicated this sensitivity for all medical applications. Which means that right now, today, every injectable vaccine you have ever received, every surgical implant, every intravenous fluid administered in a hospital was almost certainly tested for bacterial contamination using the blood of an animal whose lineage is older than the first fish with a jaw. The horseshoe crab also has 10 eyes. 2 large compound eyes on either side of its carapace, 2 simple eyes on the top, and a set of primitive photoreceptors running along its tail that appear to synchronize the animal's internal biology with the lunar cycle. This is likely why mass spawning events align with full moons in May and June. The same gravitational signal, the same beach, the same behavior, 450 million years without interruption. The Nobel Prize in Physiology or Medicine, awarded in 1967, went in part to researcher H. Keffer Hartline for work conducted on the horseshoe crab's compound eye. Specifically, for identifying the principle of lateral inhibition, the neural mechanism by which visual systems detect contrast and edges. The same principle operates in your retina right now as you watch this. A 450 million year old eye helped explain how your own eyes work. The standard evolutionary explanation is that the horseshoe crab reached a state of functional optimization so complete that natural selection found nothing to improve. Its armor, its generalist diet of worms, mollusks, and algae, its ability to tolerate a wide range of water temperatures and salinities all added up to a survival toolkit robust enough to outlast 5 separate planetary catastrophes. Geologists call animals like this stabilomorphs, organisms that found the answer and stopped asking the question. But stabilomorphy does not explain the specific texture of what survived. Trilobites were the horseshoe crab's close relatives, equally armored, equally widespread. Survivors of multiple extinctions before the Permian event finally erased every last one of them. The horseshoe crab was in the same ocean, facing the same conditions. Something different happened for one and not the other. What exactly, no one has cleanly resolved. Some researchers ask whether the horseshoe crab's copper-based blood chemistry interacts with environmental catastrophe in ways that iron-based biology does not. Whether those 10 eyes and that lunar synchronized tail represent a sensory picture of the world so fundamentally different from most animals that the horseshoe crab was, in some sense, tracking something the trilobites could not see. The archive is in the blood. We are only beginning to read it. Thousands of meters below the surface of the Pacific Ocean, where the pressure would fold steel and the water barely moves, there is a creature that belongs to no category you have been taught. It is not a squid. It is not an octopus. It is something that existed before both of those things were invented, something that occupies its own order of life with no other living members, and it survives in water so stripped of oxygen that almost nothing else in the ocean can breathe it. The vampire squid. Imagine water with almost no oxygen, not low oxygen, near zero oxygen. A band of ocean, several hundred meters deep, that wraps around the tropical and subtropical Pacific and Atlantic like an invisible suffocating belt. Most fish cannot function in it. Most invertebrates avoid it. Predators don't enter. Nothing hunts there because nothing can stay alive there long enough to hunt. For most of the ocean's residents, this is a dead zone. For the vampire squid, it is home. Vampiroteuthis infernalis, the vampire squid from hell. The name was given by German marine biologist Carl Chun, who first described the animal in 1903 following a deep-sea expedition aboard the research vessel Valdivia. Chun recognized immediately that the animal fit into neither the squid nor the octopus family. He placed it in its own order, Vampiromorphida. That order contains exactly one living species. Every other member is extinct. The vampire squid is the sole surviving representative of an evolutionary branch that diverged from both squids and octopuses somewhere between 200 and 300 million years ago, during the Carboniferous or early Permian period before the dinosaurs existed. It watched its entire family disappear. Then it went back to hanging motionless in the dark and kept going. The oxygen minimum zone it inhabits typically contains between 0.5 and 1.5 milliliters of dissolved oxygen per liter of seawater. The surface carries roughly 6 to 8 milliliters per liter. Most fish require at least 4 to survive. The vampire squid requires almost none. Its blood contains a highly specialized hemocyanin protein that binds oxygen with extraordinary efficiency at low concentrations, a biochemical adaptation so unusual that physiologists are still working to fully characterize its mechanics. It is a different relationship with oxygen than most animals have, one that seems to operate by different rules. For decades, researchers assumed the vampire squid was a predator like other cephalopods, hunting small crustaceans or fish in the darkness. The truth, which emerged from a landmark study by researchers Henk Jan Hoving and Bruce Robison published in the Proceedings of the Royal Society B in 2012, was stranger. The vampire squid does not hunt. It eats marine snow, the continuous fall of dead organic particles, fecal pellets, shed exoskeletons, and decomposing organisms that drifts down from the sunlit world above like a slow, cold rain. It is the only cephalopod known to feed this way. In an environment where hunting costs energy that cannot be replaced, the vampire squid simply opened its arms, trailed its filaments in the dark, and waited for the dead to come to it. Its eyes are proportionally the largest of any animal on Earth, approximately 2.5 centimeters in diameter on a body 30 centimeters long, which is roughly equivalent to having a volleyball-sized eye in a human face. In a zone of near-total darkness, with no sunlight penetrating from above and no bioluminescent prey to track, the question of what those enormous eyes are actually processing is one that science has not definitively answered. When threatened, the vampire squid does something no other cephalopod does. It does not release ink. It releases a cloud of bioluminescent mucus, a glowing blue mass that remains lit for up to 10 minutes, disorienting predators in a zone of the ocean where a sudden burst of light is one of the most distracting things imaginable. Individual photophores at the tip of each arm can also flash independently, potentially scrambling a predator's sense of where the animal's body begins and ends. This is not a primitive defensive mechanism. It is a sophisticated one, specifically engineered for the sensory world of an oxygen minimum zone, a world that very few scientists have ever visited and that our instruments have barely characterized. Deep-sea biologists explain the vampire squid's survival through the logic of the refuge. By colonizing a zone where almost nothing else could breathe, it removed itself from predation and competition. Its passive feeding strategy eliminated the need to pursue prey in a low-energy environment. The stable, isolated conditions of the deep oxygen minimum zone provided consistency across the mass extinctions that reset the surface world. This is a coherent argument. It is also incomplete. Oxygen minimum zones have expanded and contracted dramatically across geological time. During warming periods, they swell. During cold periods, they shrink. The vampire squid's lineage passed through the Permian extinction, the Triassic-Jurassic boundary event, and the KPG impact, all periods during which ocean chemistry fluctuated in ways that would have altered the depth, thickness, and chemistry of oxygen minimum zones significantly. The refuge moved, and every single relative this animal had went extinct. The vampire squid did not. There is no fully satisfying explanation on the table for why this one species, alone among all Vampiromorphidae, came through everything intact. Some researchers ask whether its hemocyanin biochemistry, its oxygen-binding proteins, represent a metabolic architecture so fundamentally different from iron-based biology that it responds to mass extinction conditions in ways our current models cannot predict. Others note that an animal with the largest relative eye size on Earth, in a zone of permanent near darkness, is gathering information at a sensory scale we have barely begun to imagine. What it sees in that darkness and what it has always seen, we do not know. From an animal that survives by making itself impossible to follow into the dark, we turn to one that survives by being almost impossible to ever find. It has spent 80 million years in the deepest trenches of the ocean. It has 300 teeth, a jaw that fires forward out of its skull, and a body so rarely photographed alive that for most of its evolutionary history, science did not even know it existed. The frilled shark. In January of 2007, a fisherman working waters off the coast of Shizuoka, Japan, pulled up something that made him stop the boat. It was almost 2 meters long and eel-shaped, with 6 frilled gill slits ruffled around its neck like the collar of an Elizabethan portrait. Its mouth contained 300 teeth, each one shaped like a 3-pronged trident, arranged in 25 backward-facing rows. Staff at Awashima Marine Park kept it in a shallow tank. It died within hours. The footage they recorded in that window is, for most researchers who study this animal, the clearest view of a living specimen they have ever seen. The lineage it belongs to was already ancient when the last dinosaurs were alive. This is the frilled shark, Chlamydocelicus anguineus, first formally described in 1884 by ichthyologist Samuel Garman from deep-water trawl specimens collected near Japan and Portugal. Garman recognized it immediately as something anomalous. The family it belongs to, Mitsukurinidae, was known from Cretaceous fossils approximately 125 million years old, placed in the same geological era as Tyrannosaurus rex and Triceratops. Nobody expected it to still exist. Garmin specimens proved it did. The 2007 footage proved it was still doing what the fossils suggested it had always done. Its anatomy is a catalog of features that modern sharks abandoned long ago. Almost all living sharks have 5 gill slits. The frilled shark has 6, an arrangement found in ancient hexantiform sharks and in Devonian-era fossils that predate the frilled shark's own confirmed lineage by hundreds of millions of years. Its 300 tricuspid teeth are so morphologically distinctive that paleontologists use fossil examples of individual shed teeth to map the animal's historical distribution across deep-sea sediment cores. A single tooth is enough to confirm its presence. The jaw mechanism is unlike anything else in the living shark world. Standard shark biting involves some forward protrusion of the jaws as the mouth opens, a moderate extension that enhances grip on prey. In the frilled shark, the entire jaw assembly, both upper and lower, projects forward from the skull along a separate axis, covering the distance in under 1/3 of a second, fast enough that the strike is invisible to the human eye at normal video frame rates. Researcher Kazuhiro Nakaya of Hokkaido University and colleagues documented this mechanism in a 2008 study published in the Journal of Zoology using high-speed video that finally captured what trawl fishermen had been describing for decades. The jaw does not open. It launches. Its reproductive biology may be the strangest detail of all. Female frilled sharks carry live young, and the gestation period is estimated at up to 3.5 years, extrapolated from the growth rates of embryos found in captured females. If confirmed, that is the longest pregnancy of any vertebrate animal. 3.5 years per pregnancy in a low-food deep ocean environment in a species that lives at depths between 120 and over 1,000 meters. The arithmetic of population maintenance under those conditions is genuinely difficult to reconcile with the fact that the species still exists. The flat, blade-like snout is densely covered with electroreceptive pores, the ampullae of Lorenzini, which detect the electrical fields produced by the muscle contractions of nearby animals. In total darkness, in water where light has not penetrated for millions of years, the frilled shark does not see its prey. It feels the electricity of its existence. The snout is a detector. The jaw is the trap. Both are features confirmed in the fossil record of the lineage over 100 million years ago. Deep-sea biologists explain the frilled shark's survival through ecological isolation. By retreating to deep cold water where the productive, competitive, rapidly evolving shark communities of shallower zones never followed, it kept its niche stable and its body plan uncontested. In a low-energy environment where prey is scarce and encounters are infrequent, a slingshot jaw that maximizes catch probability on rare contact makes more sense than a pursuit strategy. The explanation holds together. But a 3.5-year pregnancy in a species that lives in famine conditions makes population recovery from any catastrophic event extraordinarily slow. One disrupted generation, one oceanographic event that shifts food availability for long enough, and a population of slow-reproducing animals at depth is almost impossible to rebuild quickly. How did a species with these reproductive constraints survive extinction events that reset ocean fauna far faster than frilled shark generations can replace themselves? Some researchers suggest that the deep-sea habitat's extreme stability, its consistent cold, its pressure, its chemistry simply never reach conditions lethal enough to cause a complete population collapse. Others ask whether the animal's electroreceptive snout and its 3.5-year gestation mean it is tracking and timing its reproduction to environmental cycles we haven't measured, coordinating across deep populations in ways that trawl data, capturing a handful of individuals per decade, would never reveal. The truth is we have seen this animal alive for hours across 125 million years. What it does in the rest of that time, in the dark, is almost entirely unknown. The frilled shark survives in the dark by being impossible to catch. The next creature on this list has spent 500 million years doing almost the opposite. It is one of the most recognizable shells on any beach in the world. And the animal inside that shell watched its own family, the most successful group of ocean predators that ever lived, get wiped from the face of the earth 66 million years ago. It kept swimming. Science has never fully explained why. The Nautilus. For 250 million years, a group of cephalopods called ammonites dominated the world's oceans. They came in thousands of species. Some grew to 2 meters across. Their coiled shells were so numerous and so geographically widespread that geologists use ammonite fossils to date rock layers on every continent, the way a calendar is used to mark time. They were the benchmark of marine abundance. They were, by almost every evolutionary measure, more successful than their closest living relative, a slower, smaller, less diverse group called nautiloids. The ammonites had more species, more variety, faster reproduction, greater range. Then 66 million years ago, the asteroid arrived. Every ammonite species on Earth was gone within thousands of years. The Nautilus, the ammonites' nearest surviving cousin, was right there in the same ocean, in the same extinction event, and it did not die. Researcher Peter Ward, who has spent decades studying both fossil ammonites and living nautiluses at the University of Washington, has stated explicitly that there is no fully satisfying explanation for this differential survival. It is, in his framing, one of the most puzzling problems in invertebrate paleontology. The Nautilus lineage is 500 million years old. Its chambered shell, a planispiral coil divided into sealed gas-filled chambers connected by a central tube called the siphuncle, appears in Ordovician fossils laid down 500 million years ago and is functionally identical to the shells of animals alive in the Indo-Pacific today. It survived the Ordovician extinction, the Devonian extinction, the Permian extinction that killed 96% of all marine life, the Triassic-Jurassic event, and the KPG impact. 5 extinctions, 5 resets of ocean fauna. The Nautilus navigated everyone. The siphuncle is part of what makes the animal remarkable. By regulating the gas composition inside each of its 30-plus sealed chambers, the Nautilus controls its buoyancy with enough precision to migrate vertically over 600 meters in a single night. It descends during the day to depths where cold and darkness offer some protection, then rises toward the reef slope at night to feed on crustaceans and carrion. The mechanics of this gas exchange system, biological engineering refined across 500 million years of selection, have no direct equivalent in any other living cephalopod. Its feeding apparatus is equally anomalous. Unlike the squids and octopuses that evolved after it, the Nautilus has no suckers. Its approximately 90 tentacles grip prey through chemical adhesion alone, a contact system so different from the suction cup architecture of modern cephalopods that it represents a genuinely separate evolutionary solution to the problem of catching food. The leading hypothesis for why the Nautilus survived the KPG extinction while every ammonite died focuses on egg strategy. Ammonites are thought to have spawned small, planktonic eggs that developed near the ocean surface, a zone catastrophically disrupted by the impact winter that followed the asteroid strike, when surface productivity collapsed for years. Nautiluses are thought to have laid large, yolk-rich eggs on the seafloor, able to sustain embryos independently of surface food chains throughout the disruption. This difference, a matter of egg size and placement, may have been decisive. It is the best current explanation. Peter Ward himself notes it does not fully account for all the selectivity of the extinction, and no KPG-era Nautilus eggs have been directly compared to ammonite eggs in the same geological context. The evidence is inferential. What makes the Nautilus's survival genuinely hard to frame as a simple stroke of ecological luck is the cumulative record. The egg hypothesis addresses the KPG event. It does not address the Permian extinction, which had a completely different kill mechanism. Ocean acidification, anoxia, and temperature collapse sustained over thousands of years. It does not address the Ordovician or Devonian extinctions, each with their own distinct environmental signatures. The Nautilus survived all of them, each time by a different mechanism supposedly, or perhaps by the same one we haven't isolated yet. Some researchers ask whether the Nautilus's deep reef slope habitat, consistently occupying the zone below the productive surface layer, functioned as a buffer across multiple extinction mechanisms simultaneously. Others note that an animal capable of migrating 600 meters vertically in a single night, guided by 90 chemical-sensing tentacles and a biological gas regulation system 500 million years old, may be interacting with oceanic environmental change in ways that trawl surveys and reef edge cameras cannot fully characterize. The Nautilus is critically threatened today, not by geological catastrophe, but by the shell trade. CITES Appendix 2 protections were enacted in 2017 after surveys found that some Pacific populations had declined by more than 80% in harvested areas. An animal that outlasted every mass extinction in 500 million years is now endangered by people who want to hang its shell on a wall. From the Nautilus watching its family disappear, we move to an animal that has been almost impossible to catch even alive. It exists in a category so anatomically strange that scientists gave it a name referencing vampires and hell, not as an exaggeration, but as a straightforward description of what they were looking at. It is not a squid. It is not an octopus. It is something older than both of them, and it lives where almost nothing else can breathe. Wait, we already met the vampire squid. So from the Nautilus, we cross to something that shouldn't function without a nervous system, that has been alive as individual organisms for 11,000 years, and whose lineage has been solving the problem of coordinated animal response since before the first fish with a jaw ever existed. The glass sponge. There is an animal on the floor of the Antarctic Ocean that has no brain, no muscles, no heart, no stomach. No nervous system in any form that biologists recognized before they found it doing something that a nervous system is supposed to be required to do. If you introduce a chemical irritant to one end of this creature, a wave of electrical activity moves through its entire body within seconds and causes it to respond. Coordinated, unified, purposeful. It does this without a single neuron. And it has been doing it in the cold and darkness of the deep ocean for 600 million years. This is the glass sponge, class Hexactinellida, the oldest confirmed animal lineage on Earth that still has living representatives. Its fossils appear in early Cambrian deposits approximately 535 million years old, and the structural features that define it, a skeleton made of interlocking spicules of biologically produced silica glass, and a body organized not from individual cells, but from a single fused continuous tissue called a syncytium, are present in those ancient fossils and unchanged in every living specimen collected from Antarctic reefs today. The syncytium is the detail that stopped physiologists when they finally looked at it carefully. In every other animal, a nervous system works because individual cells, neurons, pass electrochemical signals across gaps called synapses. The speed and precision of this process is what makes coordination possible. The glass sponge has no neurons, no synapses. Its entire body is one continuous fused mass of connected cytoplasm, and electrical signals propagate through it the way ionic waves travel through a single vast cell. Researcher Sally Lays of the University of Alberta documented this mechanism in a landmark 1997 paper published in Nature, measuring electrical arrest propagation across glass sponge bodies at speeds of approximately 0.2 to 0.3 centimeters per second. Slower than nerve conduction, but functionally equivalent in outcome. The sponge coordinates, without a nervous system. It is the only animal alive that does this. The mechanism evolved once in the Ediacaran period and was never repeated in any subsequent animal lineage. Every other animal that evolved afterward built neurons instead. Individual glass sponges grow extraordinarily slowly in the cold water of the deep ocean. Growth rate modeling by researchers studying Antarctic and North Pacific populations has estimated individual organisms at over 9,000 years old, and some estimates extend beyond 11,000 years. 11,000 years. That is older than the pyramids of Giza, older than the earliest known cities, older than written language. Some of the individual glass sponges alive in the Hecate Strait off British Columbia, where a reef complex spans approximately 700 square kilometers, were already ancient when the last ice age ended and the first permanent human settlements were being established in Mesopotamia. The reef itself has been estimated at approximately 9,000 years old, a living structure assembled over a span of time that encompasses most of human civilization. The silica spicules that form the skeleton are not inert. Researchers at Bell Laboratories, comparing the optical transmission properties of glass sponge spicules with engineered fiber optic cables, found that the biological structures performed comparably to industrial fiber optics in some light transmission properties, with the added feature of being self-assembled at ambient ocean temperatures without industrial processing. The sponge builds fiber optic cables in the dark, without knowing it is doing so. Evolutionary biologists explain glass sponge survival through the logic of ecological minimalism. An organism that requires no locomotion, no active prey capture, and no complex organ systems to maintain has an extraordinarily low metabolic footprint. It filters bacteria and dissolved organic matter from seawater, an activity that can continue as long as water flows and organic particles exist, which they have for 600 million years. The deep cold water it inhabits has remained comparatively stable across geological time, providing the consistency that a sessile, slow-growing organism needs. The syncytial coordination mechanism, while scientifically unusual, is simply an alternative engineering solution that works for its purpose. But here is what that explanation leaves out. The syncytial body plan evolved once in this lineage and was never replicated by any other animal in 600 million years of evolutionary experimentation. If it is a simpler, more efficient solution to coordinated animal response, it should have appeared again. In bacteria, in other early animals, in something. Instead, every animal that evolved after the glass sponge built neurons, vastly more metabolically expensive, vastly more complex, and apparently preferable to the sponge's existing solution. Why? What does a nervous system provide that a syncytium cannot? And why did evolution pay that cost 600 million years ago and keep paying it in every animal lineage ever since, while the glass sponge, doing it differently, kept working perfectly? Some researchers ask whether the glass sponge's architecture represents not a primitive precursor to the nervous system, but a parallel solution, a genuinely different answer to the same biological problem, one that the rest of evolution passed by for reasons we haven't identified. Others note that an organism living for 11,000 years in a slowly changing ocean is accumulating environmental information across timescales that make the concept of ecological adaptation almost impossible to apply in any conventional sense. The archive is not only in fossils. Some of it is still alive, and it is still growing. From an animal that cannot move, we turn to one that can barely be bothered to. The next creature on this list survived 5 years without a single meal, in a tank, under observation, and then died of causes the post-mortem could not identify. Its lineage is 160 million years old. It has eyes calibrated for total darkness, a metabolism that biologists describe as among the most suppressed of any known crustacean, and a body the size of a football that looks, depending on your frame of reference, either like a giant pill bug or like something that has no business existing in the same world you live in. The giant isopod. In January of 2009, staff at the Toba Aquarium in Japan noticed that one of their giant isopods had stopped eating. This was not immediately alarming. The animal, cataloged simply as isopod number 1, had eaten before, and the deep sea is a place of feast and famine where long fasts are part of the biological rhythm. Staff monitored it. They offered food. The isopod declined. Weeks passed, then months, then a year, then 2 years. The animal showed no visible signs of distress. Its carapace was intact. Its behavior was normal. It moved occasionally, the way deep-sea creatures move, with the unhurried deliberateness of something that has never needed to rush. On February the 14th, 2014, 5 years and 43 days after its last recorded meal, isopod number 1 died. The postmortem examination found its body in normal physical condition. The cause of death was not determined. The giant isopod, Bathynomus giganteus, is the largest living representative of the order Isopoda, the same group that includes the small pill bugs that roll into balls in your garden. The pill bug reaches approximately 2 centimeters. The giant isopod reaches 50 centimeters, sometimes more, and can weigh over 1.5 kilograms. This phenomenon, deep-sea gigantism, the tendency for crustaceans and other invertebrates to grow to implausible sizes in the deep ocean, is documented across multiple animal groups and remains, at the mechanistic level, genuinely unexplained. The leading hypotheses involve reduced predation pressure, cold water metabolic effects, and dissolved oxygen availability, but no single model accounts for the size differential cleanly. The animal was first described by French zoologist Alphonse Milne Edwards in 1879 from a specimen dredged from the Gulf of Mexico, and its discovery was significant beyond its immediate strangeness. It helped dismantle the azoic hypothesis, the then prevailing scientific view that the deep sea was too hostile to support complex animal life. The giant isopod was direct evidence that it was not. The lineage belongs to deep-sea benthic isopoda, tracing back to Jurassic deposits approximately 160 million years old, and the order Isopoda itself extends into the Carboniferous, approximately 300 million years ago. The basic crustacean architecture of this animal predates the dinosaurs by over 100 million years. Its compound eyes contain approximately 4,000 individual facets each, calibrated for sensitivity rather than resolution. In near total darkness, at depths between 170 and over 2,000 meters, the eyes are not forming detailed images. They are collecting every available photon, building a light sensitivity profile for an environment where light functions more as a signal than a source of vision. What signals the giant isopod is tracking at depth with those eyes has never been fully mapped. The metabolism is what makes biologists stop and reconsider their frameworks. Estimated basal metabolic rates for giant isopods suggest a level of energy consumption somewhere between 1/7 and 1/10 that of a shallow water crustacean of comparable mass. This suppression partially explains fasting endurance. Partially. A metabolic rate reduced to 1/7 does not, in any simple arithmetic, produce a 5-year survival window without food. Something else is happening in the physiology of an animal that can shut itself down to near zero energy expenditure and maintain all systems in functional condition across years of complete caloric deprivation. Researchers studying this have noted that the giant isopod may enter a true torpor-like state, a metabolic dormancy that has no exact equivalent in any other studied crustacean. But the mechanism has not been characterized fully because accessing live specimens at depth for physiological study requires equipment and time that the field has not yet fully committed to this animal. When food does arrive, in the form of a whale fall or large fish carcass sinking from above, the giant isopod abandons all metabolic restraint. It gorges. Researchers observing feeding events describe animals that continue eating until they are physically incapable of moving, then lie motionless for extended periods while digestion proceeds, apparently indifferent to predation risk. This is not a measured response. It is the behavior of an animal that has spent millions of years calibrated for scarcity and recognizes abundance as a finite window that must be maximized completely before it closes. Deep-sea biologists explain the giant isopod survival through feast or famine ecology. An animal that can suppress its metabolism across years between food events and then process massive caloric loads rapidly when food arrives is perfectly adapted for an environment where whale falls might be separated by decades. The fasting endurance is not a mystery in this view. It is the point. The evolutionary explanation is coherent. But isopod number 1 did not die of starvation. After 5 years and 43 days of confirmed food deprivation with no caloric input of any kind, its body was examined and found in normal physical condition. The cause of death was not identified. Some researchers ask whether giant isopod physiology involves dormancy mechanisms that simply have no analog in the shallow water animals from which our physiological models are largely derived. Others note that an animal from a 300-million-year-old lineage living in an environment of extreme pressure, cold, and darkness may be doing things metabolically that we have barely begun to characterize because we have almost never studied one in situ, in its actual environment at actual depth. The aquarium tells us it can survive 5 years without food. The ocean has not yet told us what else it can do. The giant isopod survives in darkness by shutting itself down until the world provides something worth opening for. The next creature on this list is easy to miss entirely. It looks like a shellfish. You could find it on a beach and set it down without a second thought. But that shell is almost word for word identical to a fossil laid down 500 million years ago, and the animal inside has survived every planetary catastrophe since. It is, by some measures, the oldest living animal genus on Earth. Lingula pick up a small tongue-shaped shell on a beach in Japan or Vietnam, and you might be holding the oldest living animal genus documented in the scientific record. Not the oldest body plan, not the oldest family, the genus. The same genus, Lingula, confirmed in Cambrian marine deposits approximately 500 million years old. The same organism, or close enough that paleontologists see no diagnostic difference, was filtering water from the mud of a Cambrian sea, when there were no vertebrates, no land plants, no insects, no fish with jaws, nothing that would be recognizable as a complex animal by modern standards. 500 million years later, it is still there. Lingula is a brachiopod, one of a group of shellfish that superficially resembles a clam, but is not related to one in any meaningful way. Its 2 shells are held by a muscular stalk called a pedicle that anchors it to the seafloor and retracts the animal rapidly into its burrow when disturbed, a behavioral defense mechanism visible as trace fossils in Cambrian rock from 500 million years ago. The same retraction behavior is performed by living Lingula on beaches today. The mechanism was not updated. The animal that invented it still uses it. The case for Lingula as the oldest animal genus is specific and documented. Paleontologists comparing shell morphology, hinge structure, and pedicle geometry between Cambrian fossils and living specimens find no diagnostic difference sufficient to assign the fossils to a different genus. This is the definition of genus-level continuity across 500 million years, a span that encompasses 5 mass extinctions, multiple rearrangements of the continents, the complete rise and fall of the age of dinosaurs, the evolution of every vertebrate animal on Earth, and the entirety of human history. All of it elapsed. Lingula filtered bacteria from coastal mud and did not change. In 2015, a genome sequencing study of Lingula anatna, led by researchers at the Okinawa Institute of Science and Technology, published results in Nature that added a dimension to the mystery that body plan comparison alone could not reveal. The protein-coding genes of Lingula evolve at rates among the lowest recorded for any animal. Some gene families showed essentially no substitution over hundreds of millions of years, a finding so anomalous that it prompted immediate questions about the relationship between evolutionary rate and niche stability. But the study also found something that a simple story about frozen genetics could not accommodate. Certain gene families related to stress response and detoxification showed significant expansion in the Lingula genome. Active, ongoing molecular adaptation, the animal was evolving. just not where anyone could see it. Its visible architecture stayed identical, while its invisible biochemistry kept updating its defenses against whatever the 5 extinction events had thrown at it. The ecological picture helps clarify the survival somewhat. Lingula lives in intertidal mud flats, shallow, low-energy, organically rich coastal zones. It tolerates salinity variations, temperature swings from 5 to 30 degrees Celsius, dissolved oxygen levels near anoxic thresholds, and high sediment loads that would suffocate most marine invertebrates. This tolerance profile maps directly onto survival through events with different primary kill mechanisms. The Permian extinction depleted ocean oxygen and collapsed pH. The KPG impact disrupted surface productivity and lowered temperatures. Lingula's broad physiological tolerance meant it was never dependent enough on any single environmental parameter to collapse when that parameter shifted. But here is what that framing misses. The brachiopod phylum as a whole was extraordinarily diverse before the Permian extinction, with over 12,000 described fossil species occupying a vast range of marine environments. After the Permian, only approximately 300 to 400 species survived, a collapse from which the phylum never recovered. Most brachiopods were wiped out. Many of them inhabited environments similar to Lingula's shallow coastal zones, similar tolerances, similar body plans. Lingula survived. They did not. The ecological tolerance story explains survival in principle. It does not fully explain why this genus, specifically across every extinction event, was the one that came through. Some researchers ask whether the genome's active expansion of stress response genes represents a hidden record of each extinction event, a molecular archive of the specific chemical and thermal insults each catastrophe delivered, preserved in the DNA of an animal whose shell has looked the same since the Cambrian. Others note that an organism carrying 500 million years of unbroken survival history in its biochemistry is, in a very literal sense, a living library of how life on Earth responds to catastrophe. We have barely opened the first page. The ocean floor has been keeping something. From the Lingula's shallow mud flat, we descend for the last time into the deepest cold, to an animal so ancient that its body predates everything we recognize as vertebrate, and whose genome, when finally sequenced, turned out to contain the oldest working record of how you were built. The Chimera. In 2014, a large international team of scientists sequenced the genome of a deep-sea fish and found something inside it that required them to stop and look again. The animal was pale, translucent-skinned, with a snout shaped like a spatula, a long tapering body that trails off into a rat tail, and eyes that glow green when light hits them. It is sometimes called a ratfish, sometimes a ghost fish, sometimes a spookfish. Its formal name is the elephant shark, Calarhynchus mealy, though it is not a shark. It belongs to a class called holocephali, whose lineage diverged from the ancestors of modern sharks and rays approximately 400 million years ago, in the Devonian period. That is 165 million years before the first dinosaur. The genome that the team, led by Birappa Venkatesh of the Institute of Molecular and Cell Biology in Singapore and published in Nature in 2014, found inside this animal was unlike anything they had sequenced before. It was not just ancient, it was slow. And because it was slow, it was intact. And because it was intact, it held answers. The elephant shark genome evolves at approximately 5 to 10 times the rate of the human genome below its normal pace. This is the slowest protein evolution rate ever recorded for any jawed vertebrate. In practical terms, what this means is that the elephant shark's genome has accumulated far fewer mutations over 400 million years than most vertebrate lineages accumulate in far shorter periods. Gene sequences that faster-evolving animals modified, obscured, or overwrote are still readable in the chimera's DNA in something approaching their original form. The genome is not just old, it is preserved. The Venkatesh team used this property to do something remarkable. By analyzing the elephant shark's genome alongside those of faster-evolving vertebrates, they were able to trace the evolutionary origins of the adaptive immune system, the mechanism by which vertebrates generate antibodies and mount targeted responses to specific pathogens, further back into evolutionary history than any previous analysis had allowed. The chimera genome contained well-preserved orthologs of the RAG recombination genes, the genetic machinery that underlies antibody diversity in every jawed vertebrate, including humans. These sequences were readable in the elephant shark in a form that was no longer legible in most other sequenced vertebrates because those lineages had evolved too fast. A 400-million-year-old fish's DNA was required to explain a fundamental feature of your immune system. The chimera has no bone in its body. Its entire internal skeleton, the neurocranium, the pectoral girdle, the jaws, is made of cartilage, not as a primitive stage that will later calcify, but as a permanent architectural choice made 400 million years ago and never reversed. This places it alongside sharks and rays as one of the few major vertebrate lineages that never switched to mineralized bone, though the chimera's cartilaginous skeleton evolved independently of the sharks rather than being shared from a common ancestor. The lineage has persisted in the deep ocean across 4 mass extinctions following its divergence from early sharks in the Devonian. In 2021, a new species, Chimera caerophila, was described from specimens collected in the Northwest Pacific, indicating that the group contains more diversity in more places than previously characterized. The deep ocean, in this as in so much else, was not fully searched. Evolutionary biologists explain the chimera genome's slow evolution through a combination of factors: long generation times, low reproductive rates, stable deep-sea conditions, and the absence of the intense selection pressure that accelerates genomic change in high-population, short-lived species. The same ecological isolation that explains morphological stasis in the coelacanth or the frilled shark may, in the chimera, have extended to the genome itself, producing an organism that changes slowly at every level, not just the visible one. It is a coherent model. But a genome that evolves 10 times more slowly than the human genome over 400 million years is not just slow, it is a record. And the fact that the record proved useful, that it contained information about vertebrate evolution that faster-changing genomes had rendered illegible, raises a question that cuts across everything on this list. What if the deep ocean is not simply a refuge for animals that ran out of reasons to change? What if it is a library built from cold and darkness and pressure and geological time, stocked with biological solutions and evolutionary records that the productive, competitive, fast-changing surface world has long since overwritten? We went to that library in 2014 and found an explanation for your immune system. What else is down there that we haven't opened yet? The chimera's genome is approximately 974 megabases in size. The fraction of it that has been analyzed and understood is small. The fraction that has been connected to anything in human biology is smaller still. And the animal carrying it is alive right now in cold water, at depth, with its spatula snout pressed close to the seafloor, patient in the darkness, 400 million years old and still full of information we have not yet learned to read. 10 creatures, 600 million years of combined ancestry, 5 mass extinctions crossed and survived. And when you lay them beside each other, the coelacanth's hinged skull, the horseshoe crab's blue blood, the vampire squid's glowing mucus, the frilled shark's 3-year pregnancy in the dark, the nautilus watching every ammonite die, the glass sponge conducting electricity without a single neuron, the giant isopod outlasting 5 years without food, the frilled shark's 300 trident teeth, the lingula shell unchanged since the Cambrian, the chimera genome reading like a master record of vertebrate life itself. They stop resembling a list of anomaly. They start looking like something deliberate, not random survivors. An archive, maintained in cold water and darkness and crushing pressure, stocked with biological solutions and evolutionary records that the fast-changing surface world has long since lost or modified beyond recognition. The scientific consensus on all 10 of these animals is, in its broad form, reasonable. Deep-water refugia protect against surface extinction events. Stable niches reduce selection pressure. Generalist physiology outlasts specialist physiology. These are not bad explanations. They are incomplete ones. They explain why survival was possible. They do not explain why it was so consistent across 5 separate extinction mechanisms, across animals with completely different biologies, in a pattern so regular it almost looks like a system. What the deep ocean may be telling us is that evolution is not only a story of change, it is also a story of conservation, of answers that were found early and were worth keeping. The coelacanth's limb-like fins, the chimera genome, the glass sponge's syncytial electricity, the nautilus's chambered buoyancy control, these are not primitive leftovers waiting to be replaced. They are working tools that outlasted every alternative thrown at them by 450 million years of planetary catastrophe. And we found them not in museum cases, but alive, in the darkest places we had barely thought to look. Most people hear the phrase living fossil and picture a curiosity, something odd and distant, interesting for a moment, and then filed away. You sat with all 10 of these animals long enough to understand that they are not curiosities. They are the oldest functional archive life on Earth has produced, and the fact that you made it here means you are the kind of person who reads the archive rather than walking past it. This channel is for people who look at the standard explanation and ask what it leaves out, who understand that the most unsettling discoveries are rarely the dramatic ones. They are the quiet ones. An unnamed fish in a South African fish market. A shell in the mud that has never changed. A genome that turns out to explain how you are built. Hit subscribe and make your seat in this archive official. Then go to the comments and tell me which of these 10 animals you think is carrying the piece of information that will change everything, and what question you would ask it if you could. The floor is yours. The ocean is 4 kilometers deep in places we have never mapped. In those places, right now, there are animals carrying body plans older than the Atlantic Ocean itself, older than the Himalayas, older than almost every mountain and continent in their current form. They do not know they are ancient. They do not know they survived. They simply continue, cold and slow and patient, in a darkness we visit for hours and they have never left. The archive was never closed. Most people never find it. You just did.